Good afternoon, everybody. Welcome to our last lecture for the semester and our last week for the semester. I'm also kind of assuming that you haven't taken the final yet. Otherwise, I mean, why would you be here? But I will let you know that we're going to go over last week's exam at the end, if anybody wants to stick around for that. And we'll be answering any general questions. But I do want to say this for the record. I just sent out an email a couple of minutes ago. The final does not drop, and I don't give extensions. The due date is this Thursday, a couple of days. So please take the final today or tomorrow at the very latest. Please don't wait till Thursday to do it. If you don't take it, it will be recorded as a 0. There are no extensions. I have to turn in grades the next day. So please take care of that if you haven't already, as soon as possible after this lecture. And then whatever is in your grade book is your final grade in the class, with the only exception being if you end up with an 89.6 or something like that, right on the border, where if it rounds off to the next whole number, you get the next grade. Barring that, your grade is what it says in the grade book. Please remember there are no extra credit assignments, and I don't add points just because people really need the next higher grade. Okay, I'm going to go ahead and start the lecture, and then we will go over any past exams that you want to talk about. So let's go ahead and get started with chapter 21. Does anybody have any questions before we get started?
Can I confirm which chapters will be on the final? Because I know it says all chapters since last exam, but last week we did have a little bit questions from chapter 25. So will that be on the final?
Assume that it will. Assume that it will. So
Okay.
Just have your notes ready to go. And why? Oh, here we go.
So 23, 25, and everything from this week, correct? Okay.
Everything since the last exam, but you can also potentially assume that there may be some questions from the last couple of chapters that were on the last exam. So just in case, be ready for that. All right, can everybody hear me okay? I'm assuming so. I am on my regular Wi-Fi. This is the last day we're going to do it. Hopefully we won't get a lot of unstable internet connections, but if we do, I'll back up. Okay, we're going to go through this fairly quickly because you should have already filled in your notes. So this is just a little bit of explanation, hopefully some clarification. Okay, the immune system functions, overview of defenses. One of the things it does is scavenges dead, dying body cells. So when your cells die, your immune system gets rid of them. It protects you from pathogens and foreign molecules being anything from molecules that would come with pathogens or something even as benign seeming as a food molecule can be attacked by the immune system. Parasites are called so because they are neither bacteria nor viruses. So if it's an organism, a microscopic organism that causes disease, it's called a parasite if it's not a bacterium or a virus. So protists, fungi, and animals, examples of animals would be worms, parasitic worms. Protists, usually single-celled microorganisms, and fungi, fungus. Bacteria is in its own group, and viruses are also in their own group. And by the way, bacteria and viruses make up a big part of microbiology. By the way, if you guys are interested in taking microbiology with me this coming fall, you will need to be in the high desert because it is what is called a hybrid class. And so the labs will be on campus, but I would love to see any of you, if you still need microbiology, take that with me, and I'd love to see you face to face. All right, allergens. So I did mention this. Pollen and foreign proteins can be attacked by the immune system. Now, interestingly, allergens don't cause any disease, but we overreact to them. And that is part of the problem. That's why we get hay fever, even up to something as significant as anaphylaxis. And another thing that your immune system can do is can recognize and destroy abnormal, such as cancerous cells. And that's a focus of a lot of cancer research now. Immunotherapy, it's called, the idea that you take immune cells out, train them to attack cancer cells, put them back in, and then you cure yourself of cancer rather than needing something as barbaric as radiation or toxic chemicals. All right, immune system pathology. So if your immune system doesn't work correctly, one of the things that can be is an incorrect response. And an autoimmune disorder is what we call an incorrect response. That is, you attack something that's yourself and not a foreign invader. One example of that is lupus. I know someone with lupus. Type 1 diabetes can be caused by this, and rheumatoid arthritis is another example. Rheumatoid arthritis is caused by the fact that your white blood cells attack the tissues in the joints, causing them to become inflamed. And that's one of the reasons why there are certain medicines that to treat rheumatoid arthritis, one of the warnings of it is if you have a depressed immune system, to not use, because what it does is it basically pushes your immune system down so that it doesn't attack your own body cells. Overactive response, out of proportion to the threat. So allergies. So allergies happen because our body believes that it's a invading microorganism and it responds in that way. But just to let you guys know, pollen grains don't cause any trouble other than the fact that our body overreacts to them. Pollen grains can't cause any disease. So what happens is we overreact to it. So what we take are what are called antihistamines, because what happens is in an allergic response, histamines are released from mast cells. That causes inflammation, irritation, etc. So antihistamines block those histamines. So hay fever is minor, anaphylaxis is major. So if you have a really severe overreaction, you can get anaphylaxis, which would lead to anaphylactic shock. So a lack of response is another problem. That is the failure of the immune system to work properly. And so an immunodeficiency disease. So it can either be inherited or acquired. So for example, AIDS is acquired immunodeficiency syndrome due to a virus. So bubble boy syndrome is what you would be born with. That is the lack of an immune system, and AIDS is acquired. Okay, so some pathogens that could make you ill. What is the structure of a bacteria? It's almost always composed of cells with cell walls. Now, cell walls of bacteria are different than the cell walls of plants. And remember, humans and other animals don't have cell walls at all. We have cell membranes. They may also possess a capsule, which is a protective layer that helps it avoid the immune system or to stick to surfaces. So capsules can be around bacteria that prevents the immune system from recognizing them as a pathogen and allowing them to continue to live inside the body. What are the living conditions? Well, most can survive outside of a host cell. One of the things that we're going to be talking about is viruses need to infect a living cell and convert it into a virus-producing factory. Bacteria need a food source, but they don't need to live inside of a cell. Many live inside of organisms. They generally don't live inside of cells. Susceptibility to drugs, most can be killed by antibiotics. Now, there's a whole thing that we're going to be talking about in microbiology about antibiotic resistance. You guys have probably heard of that. MRSA, multiple resistant Staph aureus. A lot of bacteria have become resistant to antibiotics, and it's due to overprescription and improper consumption of antibiotics that has led to that. Okay, viruses. The structure of viruses is different. They are not made out of cells, and so therefore don't have a cell wall. They're composed of a nucleic acid surrounded by a protein coat. That protein coat is called a capsid. The nucleic acid can either be DNA or RNA. If it's RNA, it usually has an enzyme called reverse transcriptase that causes the cell in which it infects to produce DNA, which then takes over the cell. What are the living conditions? Most cannot survive outside of a host cell. Now, it doesn't mean they're necessarily going to die if they're outside a host cell. Many do, but it means that they can't reproduce. Must have a host cell to reproduce. So they take over the machinery of the host cell to create more viral particles. Susceptibility to drugs. They cannot be killed by antibiotics, but may be inhibited by antiviral drugs. So if you go to the doctor with a viral disease and they prescribe an antibiotic, it's not proper because the antibiotic does nothing to kill the viruses. Now, often doctors will say, Well, I'm giving it to you to suppress the bacteria in your body so that your immune system can fight the viruses better. I think that's bogus. They just want to give you something that you can go put in your mouth, take it as some kind of a thing to make you feel better. For most people who have a viral disease, it's just time and plenty of water and rest. But they often will prescribe an antibiotic because the patients really want it and feel that that's what they are entitled to. Okay, what are some body defenses against pathogens? Well, physical barriers, believe it or not, are the most important. Having an intact skin. And we're supposed to all take a break in our skin is very, very important. We wash it, we put antibiotic on it, we put a bandage on it. All those things help prevent infection. Epithelial linings and the cilia. Cilia are interesting because they can actually move bacteria on the surface away from certain areas. Okay, chemicals we find in our bodies, acids. For example, the female reproductive tract is acidic, killing bacteria that would normally live there. Mucus and lysozymes are found in the most of the mucous membranes, especially in the digestive tract. Lysozyme is an enzyme that kills bacteria. Internal defenses, immune defenses, innate, non-specific, means that it's going to react. It is not something you have to learn, and it's not something specific to a particular microorganism. So for example, inflammation. When you get inflammation as a result of an infection, what that's doing is to increase the movement of white blood cells to the site of the infection and reduce bacterial motility, which is the ability of the bacteria to swim away and infect other areas. So inflammation is an innate defense mechanism. That means it's built in. It's not based on any exposure you have. Now, acquired is when a specific pathogen, the antigen, which is a protein on the surface of the pathogen, you've been exposed. Now, you can either be exposed by the disease or by a vaccination. Either of those are. What was the last thing you guys heard me say? Anybody?
Starting with acquired.
Okay. Acquired means that you have been exposed to a particular pathogen, the antigen, that is the protein on the surface of the pathogen, and your body then has the ability to fight it because it has seen chemically the surface of this organism. Now, this can either be as a result of being ill, where you actually fight the pathogen itself, or a vaccination. A vaccination is still acquired and it's still active, but it's basically giving your body the ability to recognize it without really getting sick from it. Okay, what are some of the steps in the immune defense? Well, you either detect the invader or the foreign cells, usually by use of antibodies. The antibodies stick to the surface, and most cells that have antibodies stuck to it are labeled as invaders and destroyed by the immune system. So when this happens, the T cells are primarily what are responsible for communicating the alarm, and that is, it alerts the rest of the immune system to go after the cells that have been identified and basically pass around the antigenic protein and say, this is what you're looking for. And so then it can either suppress or destroy the invader. And one of the main ways that it does that is by using phagocytic cells. These are white blood cells that surround, engulf, and digest any invading microorganism. Okay, lymphatic system, overview of immune defense organs and cells. Now, we talked about in a recent chapter how the lymphatic system helps return leaked fluid from your capillaries back to your venous side of your blood system, but it also has an immune function, and that is bone marrow and thymus are a big part of your immune system. And they form and mature immune cells, and that's one of the reasons why they're called T cells, because they come from thymus derived. One of the things your thymus does is it helps mature white blood cells, make them better able to fight against infections. The lymph nodes are also very important because they filter lymphatic fluid. We talked in the chapter on lymph that it's a better fluid to filter because it's not full of chunks like you would find in blood. It's very clear, and anything in there potentially is something that the body could use to identify an organism upstream and then go find it and destroy it. The spleen contains immune cells that monitor pathogens in the blood. Now, you don't need to have your spleen, but it's helpful. It does help you monitor pathogens in your blood because your lymph nodes can't monitor the blood. They only monitor the lymphatic fluid. Lymphatic vessels conduct lymph to the lymph nodes, and the leukocytes are the white blood cells. Okay, this is a figure directly from your book, and this shows you your entire immune defense system, especially the organs associated with it. You see lymphatic vessels, lymph nodes, bone marrow, a blow up of the lymph nodes themselves, the thymus where the T cells are derived. Adenoids and tonsils are also involved. That's one of the reasons why doctors aren't as quick to remove tonsils as they used to be, because they have found they are useful in the immune system. All right, lymphocytes are a type of white blood cell. They're key to acquired immune response. They're usually found in lymphatic tissue. Now, lymphocytes are called that because they exist in the lymph nodes or lymphatic system. And they're the key to acquired response because they identify microorganisms or pathogens, and then often remember what they've seen, and they're able to find it again more quickly the next time it invades. Basophils and mast cells release histamines and heparin. Remember, in heparin, we use heparin as a blood thinner, and that's a term that I really hate, even though I said it. What it does is an anticoagulant. Cytokines are involved in pain and inflammation. Remember, mast cells release histamines, which also cause inflammation. And basophils, I'll be talking more about these cells later. Okay, monocytes are the very large white blood cells that mature into macrophages. And these macrophages then are going to patrol the body tissues and phagocytize or swallow and destroy old red blood cells and other damaged tissues. So these are primarily getting rid of your own damaged cells, not so much pathogens, but they can also destroy pathogenic cells. Neutrophils are the most abundant type of white blood cell, approximately 50 to 70% of the total white blood cell count, and they phagocytize invaders and release cytokines. Now, one of the types of cytokines produce fever. Now, you might say, well, what's the benefit of the fever? What the fever does is it actually causes everything to speed up. It can also partially denature bacterial and viral proteins. And fever is a natural thing. So the idea is you don't necessarily need to suppress a fever as long as it's below 103. So if you had a fever of 101, 102, that's your body's natural reaction to it. Yes, it should be monitored, but doesn't necessarily need to be suppressed because that fever is helping you fight the infection, believe it or not. Eosinophils fight parasites and contribute to allergic reactions. Now, this is interesting because our ancestors all had parasites, and humans had parasites up until fairly recently. With the advent of better sanitation and food preparation mechanisms, we don't really have it anymore, but we still have these eosinophils, which are white blood cells. So what happens is they tend to overreact when they find an allergen, such as a pollen grain. Now, they did a little bit of research, and I don't know if this is in your notes. I don't know if it was an extra credit reading or something you guys got earlier, but did you guys get a chance to read about on parasites and pollens? Was that in any of your notes? Anybody?
No.
It wasn't. Okay, well, let me tell you a little bit about the
We
research.
did have one on parasites from Africa.
Well, the thing that I was hoping that you guys had seen was called on parasites and pollens. Now, let me tell you a little bit about the research and I'll move on. What they did is they found a group of people, Aboriginal people in the South Pacific, who still had intestinal parasites because they were eating improperly cooked meat. And one of the ways you can get improperly cooked meat is by putting it in the ground in a thing called a deep pit barbecue. You guys may have heard of that. Well, some people, they don't cook their meat at high enough temperature or long enough, and they're still parasitic microorganisms, especially the like the little cysts that you would find in it. Well, these people had parasites. What they discovered when they found these people is they had almost no allergies to pollen. So when they went in there, they gave them medicines to help them fight against these parasitic worms. And guess what happened to them after they were cured of the parasitic worms? What do you think they developed?
Allergies.
Allergies. So what that tells you is these IgEs comes from eosinophil, is basically an overreaction. They're trying to find parasitic creatures such as worms, and when they can't find it, they go after the pollen grains, needing something to fight. So it's kind of interesting. If you do a quick internet search on parasites and pollens, you'll find that article. It's fairly old now, but it's still very useful, and you can read all about that. Okay, macrophages and acquired immunity. They ingest molecular or cellular antigens and digest them. So you can see an antigen. This could either be a virus, a bacterium, or anything else. So often these are sensed and engulfed. But what you notice here is once the lysosome, which is this structure here, fuses with it, breaks it down into its component proteins. Now the proteins aren't always broken all the way down into individual amino acids. Some of the pathogenic proteins or antigens, they would be called, instead of being broken all the way down to individual amino acids, are presented on the surface basically to show other white blood cells. Look at what I found, and you should be on the lookout for these same pathogenic cells, because it's when you see one, kill it immediately. So the fragments of the antigen are presented on the surface of the cell, and it alerts other immune cells to this organism. That makes the immune response happen more quickly and efficiently after the initial exposure. Okay, innate immunity. As I mentioned before, physical and chemical barriers such as your intact skin. One of the reasons why skin is such an effective thing is because it's covered with oil for the most part, and bacteria and viruses hate oil. So having oil on your skin helps prevent bacterial infection, believe it or not. Mucous membranes also reduce bacterial infection. Ciliated epithelium move them away from areas that could potentially become infected. Hydrochloric acid kills bacteria in your stomach, and lysozymes, in tears and saliva also kill bacteria. Phagocytosis is macrophages, neutrophils, and NK cells. That stands for natural killer cells, are all involved in finding, swallowing, and destroying microorganisms. They engulf and digest recognized foreign cells and molecules. Now remember, if you do this against your own cells, that could be autoimmune disorder. So you hopefully don't attack your own body. The inflammatory response is also part of an innate response. I talked about this earlier. It reduces bacterial mobility. It's a physical barrier to the spread. So it's basically keeping the bacteria where they are, preventing them from swimming to new areas and spreading the infection. It can also promote tissue healing due to increased blood flow. Interestingly, the increased blood flow causes the inflammation, but the inflammation also causes the bacteria to not move. So it's kind of a catch-22, but in a positive way. This also attracts immune cells because now the immune cells can fit between the gaps in the endothelial cells of the capillaries of the tissue that's got the problem. Okay, so pathogen can bind directly to the phagocyte receptors, or you can have an opsonin molecule, which is a type of antibody. It sticks to the invading microorganism, and then any cell which is covered with these opsonin or other antibody then would be identified by a phagocyte or a white blood cell and destroyed. Okay, cytokines and signal initiation. So we talked about histamines. Mast cells produce them. It causes swelling, edema, and blood vessel dilation. So remember, the swelling is important to reduce bacterial motility, helps white blood cells get into the infected area. And what could be some problems? Well, if you have too much inflammation, it can lead to problems with blood flow. It's painful. And if it overreacts, you can get, let's see, what am I thinking of? Overreaction of the immune system would be, my brain just blanked. We were just talking about it. Anaphylaxis. So an overreaction to histamines. Now, one of the reasons why anaphylaxis causes a problem is the histamines causes blood vessel dilation. That causes a drop in blood pressure. Too much blood pressure drop can reduce cause fever. We talked about fever is good in helping speed up the reaction.
Can you repeat that thing, because you cut out for a little bit?
What was the last thing you heard me say?
The problems about the infection site, and you were saying that one of the problems is too much of a response can cause anaphylaxis, and then you were saying something.
Right. Okay, so the anaphylaxis causes blood vessel dilation. The blood vessel dilation causes drop in blood pressure. If you drop blood pressure too much, you don't get enough perfusion of blood, not enough oxygen, and the tissue can die. Or you can, you can die from too low blood pressure because your brain doesn't get enough oxygen, nor does your heart. So that's another problem associated there. Now, these interleukins, we talked about fever being good as long as it's below 103. Increase in blood vessel gaps, that allows more white blood cells and antibodies to get to the infection site. So this is one of the job of the interleukins. Bradykinins causes swelling and pain. So you might say, how could swelling and pain ever be beneficial? Let me tell you something about why pain is good. Now, I'm not one of those types, but biologically, physiologically, pain, what it says, the first thing it says is there's a, someone said is cellulite due to inflammation as well. Yes, it is due to inflammation. Okay, so what is the benefit of pain? Well, pain says, hey, you've got a problem with this body part. Don't use it because it needs to heal. Continued pain is beneficial because it means that the injury is still there and you still need to stay off of it or don't use it. The third benefit to pain, believe it or not, is it teaches you a lesson. Your body says, hey, dummy, don't let that happen again. Remember how much that hurt? So try to avoid what caused the pain before. Now, an exception to that are pain, sources of pain that we choose. For example, childbirth. Although we don't choose it specifically, biologically, we're choosing it, and things such as piercings and tattoos don't give us that, oh my God, I better never do this again, because you've chosen that activity knowing it was going to hurt. So it doesn't give you the lesson that you should never do this again. All right, acquired immune responses. So antigen-specific responses in which the body recognizes a particular foreign substance and reacts to it. So this is acquired immunity. That means you've been exposed to something, your body then reacts to it. This is mediated by the lymphocytes, which reside primarily in your lymph nodes. So beta lymphocytes or B lymphocytes, when activated, become plasma cells and begin to produce antibodies. So they basically go through a process called clonal expansion. So when one gets activated, they start to rapidly divide and they produce tons of antibodies so that those antibodies will then find the invading microorganisms, stick to them, and anything that's covered with antibodies generally is going to be destroyed. So T lymphocytes, derived by the thymus, regulate the activity of other immune cells. These are the organizers of the immune response. Natural killer cells recognize and destroy virus-infected or tumor cells. So these natural killer cells, one of their main jobs is to get rid of your own body cells that are not working properly. Different types of acquired immunity. So it can be active immunity, occurs when the body is exposed to a pathogen. It creates its own antibodies. Now, this can either be with a vaccination or with disease. It's still acquired because it came into you. Passive immunity means you get the antibodies directly from another person. So, for example, when a baby consumes colostrum or mother's milk, the antibodies come directly through the wall of the GI tract into the blood, giving them passive immunity, which means they didn't have to produce the antibodies. Somebody else produced it for them. So if you get antibodies from somebody else medically, that's also a form of passive immunity. But active immunity means you've been exposed either naturally or due to a vaccination, and you produce your own antibodies.
So is that an interferon? That's an active immunity kind of component?
Well, it's not, well, believe it or not, it's not immunity at all. Interferon is basically a drug that inhibits viral replication. So it's not really a form of immunity, because immunity comes after you've been exposed to something and your own body becomes aware of it and ready for it. So interferons aren't even a type of immunity. They're just a treatment against the particular virus that is in you now, so that you can beat it and then eventually learn from it and not get sick again. Okay, we've got to keep moving. Clonal expansion of memory. What is the clonal expansion? It's the division of cells that have been exposed to an antigen from a pathogen to produce a large number of identical cells, each with the ability to recognize the same invader. So it's basically a cloning. That's why they call it clonal expansion, because that individual that has been exposed then divides. Now you have lots and lots of those cells that can do that. Naive lymphocytes are lymphocytes not yet exposed to the antigen. One of the review questions says, do naive lymphocytes produce
Clones.
What's that?
Your question says, do they produce clones?
Well, it's kind of a complex question, because once they produce clones, they're no longer naive. But so it's kind of it's a little bit of a tricky question. I'm sorry about that, but
I was actually going to ask you about that, because I was like, how do we answer this?
That one is, I'm not exactly sure what I was thinking when I wrote that originally, but I think I'm going to go with true, because maybe it was false. I just can't remember, because it's very unclear whether I was saying they can't be naive if they start producing the antibodies or start dividing, because they'd no longer be naive. So that's one of those ones where I don't know exactly what I was thinking when I wrote it. So that's one of those times where I can't really help you with that particular
Does
review
that mean
question.
it's not going to be on the test?
I hope it's not on the final. I really hope it's not on the final. But if you take the final and you see it there and you get it wrong, send me an email and I may be able to adjust your score a little bit. But please remember, a point or two, unless you're right on the border between two grades, is not going to significantly affect your grade. And guys, if you finish the class with a 94, please don't send me any questions wanting a 95 or a 96. That's really kind of irritating. You're going to get your A. It's not going to be an A+. So even if you missed a few points and you think you deserve more, if you finish the class with anything above a 90, you're done. Take it as a win and go and just enjoy the rest of your summer. Okay, moving on. So naive lymphocytes undergo clonal expansion after exposure to destroy the invader. So once they get exposed, they're no longer naive, and then they undergo clonal expansion. So that's where the unclarity comes from. So I think I was telling my students that's true because of this particular bullet. Memory cells can remember the first exposure and continue to divide, passing on the ability and producing antibodies to that. So that's why you don't need to get vaccinated against MMR your whole life. The memory cells remember. And so if you get exposed later on, you can have a really quick response rather than a slow response. So the subsequent exposure causes the clones to divide much more rapidly. They're ready for a quicker immune response. So theoretically, yes, you would get sick, but it would be such a minor illness, you wouldn't even know you had been sick because it reacts so quickly to that re-exposure. Now, a little bit different is some viral diseases, especially coronavirus. You can get sick several times because it mutates and your body's not ready for the new mutations. So that's why I always tell people, just because you got sick from COVID-19 doesn't mean you can't get sick again. But it is true that the second time you get sick, you're probably going to get less sick because you do have some basic immunity. But I hope I don't need to tell you guys that vaccinations are far, far better than getting sick with COVID. And there's some very anti-scientific folks out there who think that there's something wrong with the vaccination. There's no solid documented evidence that there's anything wrong with any of these vaccines against COVID and to save lives. So I'm not going to get on a soapbox, but please don't start attacking science because you don't understand it. And that's mostly what I see these folks doing.
So
All
does
right,
the MMR virus not mutate? Is that why you only need one?
Yeah, well, it does mutate, but it doesn't mutate enough so that the vaccination can't get it. It just turns out that coronavirus, can mutate so quickly that you can get sick from it several times. And that's why really what they say is the best way is to be vaccinated and have gotten sick, because then you have naturally derived antibodies and memory cells from the actual infection, and you have immunity from the vaccination. And those people almost never get very, very ill. But yes, so most viruses, if you get a vaccination once as a child, don't mutate enough so that you would then have to do yearly. It looks like COVID-19 is going to be a yearly thing, in the same way that the influenza or flu virus needs a yearly update. I think we're going to be getting COVID-19 yearly updates because of the way it spread and because of its preponderance, its proclivity to evolve and mutate. Okay, so this shows you how the primary immune response works. So we have your short-lived effector cells and then the long-lived memory cells. And then when you get exposed again, there's a really quick response, so you're not even going to know you've been sick. Okay, that is our chapter. It's a little bit brief, but you guys get the idea. And so what we're going to do now is we're going to jump over to 27. Let me go and close this. Let's see. Okay. Let me share my screen from chapter 27. Here it is. Okay, I'm going to go through this pretty quickly because your notes are already filled in. All right, let's talk about sex determination. And then at the end, there'll be questions talking about upcoming final or past tests or any other questions that you have specifically. Hey, folks, quick request, if I forget to stop the recording, please remind me to stop the recording so that my questions with you don't end up on the recording for everybody to listen to. All right, so gonads. Turns out that both males and females have gonads, not just men. They're organs that produce gametes. Both men and women have them. But male gonads are testes. They produce sperm cells. Females' ovaries produce ova, not eggs. Most people still call them eggs, but the better term is ova or oocyte. Chromosomes determine gender. This is not always the case for all animals. It turns out alligators, their gender is determined by the temperature at which the eggs grow. So the ones near the top of the nest tend to be warmer, so they turn out female. The cooler ones at the bottom tend to be male. So the way that the alligator produces the nest can determine the gender ratios. But in humans, it's generally chromosome dependent. So the autosomes are all the non-sex chromosomes. There's 22 pairs of them, and the sex chromosomes. So the reason why 23andMe is called that, because you have 23 pairs of chromosomes that can be analyzed, and they can find out things about you, where you're from, who you're related to, and potentially any genetic abnormalities. So you have 23 pairs or 46 total chromosomes. Now we're going to be talking about some people who don't have 23 pairs or 46. Some people have 47, and we'll talk about what is the effect of that. Now, sex chromosomes. Females have 2 X chromosomes. The denotation is XX. One is inherited from the mother, the other from the father. So the father has an X and a Y. He gives his X to his daughter, so does the mom. Males inherit 1 X from the mother only, and the Y comes from the father. That's why males are male, because they get the Y from the dad. It says fathers determine the gender of offspring, but that only means biologically. It means which sperm cell comes into the ovum, whether it be an X bearing or a Y bearing. So that's what that statement means. Abnormal combinations, sometimes this is called super female. They do appear female. They do have problems. XXY is male appearing. So the presence of the Y basically overrides the second X. X blank is female. So this is called female. One of these has a disorder associated with it, but Turner syndrome, I want to say, is X blank. But notice here at the end, blank Y dies. What does that tell you about the importance of having at least 1 X? Well, the X is not just sex-related things. It's a whole bunch of body structures on that X. So if you don't have at least 1 X, you can't survive. So you don't need a Y, but you do need to have at least 1 X. Sex-linked traits. I talk about this a lot in my general biology class. We'll talk about it briefly here. Most human traits are located on the autosomes. That means the non-sex chromosomes, making them not sex-linked. Humans are diploid, meaning they have 2 genes for a single trait. That is, you got a full set from your mom and a full set from your dad. So you get 1 letter from each parent to indicate your genotype, or the genes that you have for a particular characteristic. Many traits are located on the X chromosome. If it is on the X chromosome, it is called sex-linked. So males show it more often because they don't have a second X. So the idea is, if a woman has a recessive trait on one of her X's for a problem, her other X is probably going to have the normal dominant gene, and she won't know she's carrying it. Since males don't have a second X, if they get that recessive problem, they will show it automatically because they are not diploid for sex-linked traits. If a male has 1 copy of recessive gene, it will show, since he has no other gene to mask it. For example, men tend to show color blindness, which is a sex-linked recessive trait, far more than women do, because women often have the normal gene hiding the sex-linked gene. But if a man gets the sex-linked gene for color blindness, he has no second X to cover it, so he shows color blindness. Females have a chance to mask a sex-linked trait with their second X chromosome and therefore not show it. Female shows sex-linked traits half and often as often as do men. Just to clarify this, you can use this on the test, but you guys know the difference between X and X squared. It's not half, is it? Right? It's exponential. So females show it exponentially less than males, but because it's written here, you can go ahead and use it for the final exam. Examples of sex-linked traits are baldness, that is male pattern baldness, hemophilia, the inability to clot your blood, and color blindness, red-green color blindness. Okay, fetal development. Reproductive structures begin to develop at about the 7th week of fetal development. There's no extra credit on the final, so don't worry about that. In the absence of male hormones, external genitalia appear female. So here's what's interesting, folks. All humans are females unless the Y chromosome turns on certain genes to produce male hormones, and that's the only people that become male. So it turns out that males are highly modified females. So everybody is female unless they have the Y chromosome, which turns them from female into male. Interesting. Gametes. The ova or oocytes, which we call eggs, are the largest cells in the human body. Why do they need to be so big? Well, it's because it's released and then it has to spend a lot of time going from the ovaries all the way down to the uterus. And in that time, they have to be dividing if they've been fertilized, and they don't get any help from the woman's body. So they have to be really large, containing lots of food for those divisions. The ova contain all the organelles that are found in the developing zygote. So here's another interesting factoid. All of your cellular organelles, especially mitochondria, are exact copies of your mother's. And it turns out that you have the same DNA in your mitochondrial DNA as your mom did, because the only thing you ever got from your dad was chromosomes, not any organelles. So you can trace your history. And that's one of the things they do at 23andMe, the history of your genetic pathway by looking at the mitochondrial DNA. Ova contain all the food necessary for the beginning of development. Remember, from the time that they're released until the time that they embed in the ovarian wall, uterine wall, the uterine wall, they are on their own to provide the nutrients that they need. Sperm cells are much smaller, containing our chromosomes only. So the way you can think about them is they're much more like torpedoes. They have to go quickly, they have to deliver something very important, but they don't have to be large because they only have to have chromosomes. Sperm cells have flagella for swimming to the ovum during fertilization. And interestingly, there is actually fluid flow going from the ovaries down to the vagina. And so the sperm cells have to swim upstream, very much like salmon swim upstream to mate. Production of gametes. Women are born with all the ova they will ever have. Interesting, the ova they get were in there when they were inside their mother. So here's an interesting thing, ladies. It turns out that the ova that are going to end up being your kids were affected by your mom, their kid's grandmother, because you were in there and all the eggs were there and being affected by what your mom was doing. So women can have an effect on their grandchildren by affecting the ova that are in their female children's body. Now, that's not the case for men because they produce sperm cells spontaneously and constantly, but there's a whole lot I could talk to you guys about if we had a longer class, but I'm just going to go ahead and move on. Women release ova between the ages of 12 and 50. Most girls start somewhere between 12, 15. 10 and 15. 12 is the most common, and 50 is a common time for the end of menses. After that time, you go through menopause. Most women go through approximately at the age of 50. Some women much, much later. There have been moms at age 60 or 65, but that's very rare. Males produce sperm continuously from the beginning of sexual maturity until death. Is it fair? No, nothing's ever really fair when it comes to gender, but it's different. It does turn out that sperm cell production diminishes with diminishing testosterone level, and that starts to diminish after about the age of 40. And so even males that are 100 years old are producing some sperm cells, but usually not enough to become fathers. Some decline in numbers at later ages. That's mostly due to diminishment of the testosterone level. Gametogenesis, gamete is sperm or egg. Genesis means to create or to grow. Gametogenesis is the development of gametes. Males produce sperm through continuous meiotic divisions, to end to end, so you're cutting the total number of chromosomes in half. In female ova, the first stages of meiosis begin during puberty, but do not complete until fertilization. So interestingly, ladies, if you've never been pregnant, you've never completed a full round of meiosis before. Every 28 days, approximately, an ovum is released. That's ovulation. Interestingly, the cycle is not the same as a calendar, which goes between 28 and 31 days, but it does tend to match the lunar cycle. So anyway, for whatever that's worth. If no sperm are present, meiosis is not completed. So if you never have sperm reaching the ovum, you never finish meiosis. Okay, so this is a figure showing how spermatogonia becomes primary spermatocyte. You get a secondary spermatocyte due to meiosis, and then eventually sperm cells are formed. Differently in females, you get a primary oocyte. These polar bodies don't become separate cells. They just disintegrate, leaving a nice full-size cell. The second polar body separates and disintegrates after fertilization, after implantation. And then you get a zygote, which is potentially going to become a baby. Control of reproduction. Hormones that control reproduction are produced in the brain. So the brain is ultimately responsible for reproduction. Gonadotropin releasing hormone, abbreviated GRH, is produced by the hypothalamus, which is the bottom part of your front of your brain, causes the anterior pituitary to release several secondary hormones. We talked about this in our chapter on hormones. Follicle stimulating hormone, or FSH, controls gametogenesis along with the sex hormones. Controls the production of gametes.
Will you repeat
Luteinizing-
that you cut out?
Well, instead of repeating it, it's all right there on your screen. I'm not saying anything that is not written there. So I'm just basically explaining, reading this. Sex hormones, androgens and estrogens are produced in both sexes due to luteinizing hormones. So this is a test question. Is it true that both men and women produce both estrogens and androgens? The answer is true. Both genders produce both types. Now, androgens tend to dominate males, estrogens in females most often. So high levels of testosterone can cause premature baldness and certain types of male aggression. Estrogens predominate in females. High levels can potentially lead to ovarian cancer, and or if you add estrogens postmenopausally, that can increase the likelihood of ovarian cancer. This shows you a figure of how sperm cells are produced. I'm not going to go through a lot of detail on this because it's not on your test. A little bit about male reproduction, the pathway of sperm cells. Hey folks, pretty much what I am going to be doing with this is more or less just reading what it says. So for the sake of time and because I have some things that I need to take care of, I'm going to go ahead and stop there. All of your notes are filled in. There's not a whole lot that needs to be explained. So at this point, what I'm going to do is we're going to start doing our review for the final. And then afterwards, I'll stop the recording. We'll go over past exams, including the final if you've already taken it, and talk about any concerns you have about any questions. So what's our first chapter that we haven't already done a review session for? Anybody have it? Is it 25?
Well, I know on the last exam, we were tested on the renal system and we had digestion, but that we were grouped in the same chapter, but we didn't do digestion. So I'm assuming digestion will be on the final.
Yes.
And all of,
Should be.
okay. And everything else that we're doing right now. So I guess,
Yes.
yeah, 25.
Okay, so the chapter on digestion is what?
23.
23. Okay, so if you guys could pull out chapter 23 and find any review questions that you had trouble finding, or you just want to double check to make sure you have the correct answer. Who would like to start us off with chapter 23?
Gosh.
Does anybody have any questions about 23?
Not really for me.
Okay, hearing none, let's jump to the next chapter. What's our next chapter after 23?
21?
21? Okay, if you did chapter 21 in advance and found some review questions that you were having difficulty with, let's go through those. Remember that one about naive lymphocytes we've already covered. So I think I'm going to go, if you have that question, I'm going to go with true on that. And hopefully that's the correct answer, if it does come up. Anybody else on chapter 21, immunity?
Can we do 34?
34, read it, please.
What is the name of the cell type that releases pyrogens? And I wasn't sure if it was supposed to be cytokines or if it was supposed to be neutrophils.
Because cytokines is a chemical. The neutrophils are the cell type.
Oh,
Okay. Yeah, cytokines are is a type of chemical, not a cell. So
okay.
I hope that helps.
Yeah, thank you.
Okay, you're welcome. Anybody else for this chapter?
Could I get help with 52?
Louder, please.
Okay, with 52, how is the
Good.
response of the immune system different after a second exposure to a pathogen than it was the first time?
Okay, so what happens after you've been exposed and you've beaten that infection? What happens the second time around? I lectured on it today.
You have antibodies, so they recognize it right away?
Well, remember, you also have memory cells. They're going to undergo clonal expansion far more quickly. So yes, the presence of antibodies, but I think that because it's a more complex question, will probably not be on your final because it would require a very complex set of answers. So I don't believe that one is on the final, if that helps.
Okay, thank you.
You're welcome. Anybody else for this chapter?
Number 19.
19, read it.
Interferon is an example of what type of drug? Is that
Okay.
a man-made vaccine or,
No, it's not a vaccine.
oh shoot, okay. An
Anybody
I
have an
know, I know it has to do with antiviral stuff, but
The antiviral is the correct answer. Antiviral. Interferon is antiviral. That's the answer I was looking for.
oh, okay, thanks. Thanks for clarity on that.
Well, that's why we do these reviews, so you can get some clarity.
Yay.
Yay. All right, anybody else? On this chapter, immunity.
Can we do 32? Not
Read it.
32, but number 30. What is heparin used for in the body in the absence of doctors?
So
Is it
it's an anticoagulant. An anticoagulant. And so it is used also in hep tubes or heparin tubes, but it does provide an anticoagulant effect inside the body.
Oh, okay, thanks.
Okay. What else? Any others on this chapter?
I have one more. Sorry.
What? No, it's okay. That's what we're here for. What's the number?
39. How is the effect of histamines both a problem and a solution in the problem?
Okay. Do you guys remember me talking about histamines causing inflammation and pain? And
Oh,
it's
so it's
so-
the anaphylaxis? Is that
so the serious problem with it is if it's overdone, it can lead to anaphylaxis. It's painful. There's swelling, discomfort, but it also, remember, it also helps you fight the infection faster. So again, that's a kind of more complex answer, not a simple answer, but so it probably won't be on the final. But remember, we talked about the catch-22 of inflammation. It's beneficial in some ways and non-beneficial in others. It can lead to decreased blood flow potentially and potentially anaphylaxis. So there's that. All right, who would like to go? Is there any more on this chapter 21? Okay, hearing none, let's go to the last chapter, which is, what do we have? 20, what is it, 27?
yeah.
27. Any of the review questions from chapter 27, you just need to let me know which section it's in so everybody else can also know. Anybody have any review question from chapter 27 that they were working on and need some clarification?
Number 12 for 27A.
Okay, read it.
Pseudohermaphrodites have both male and female organs fully developed as children. True or false?
Well, that's false for two separate reasons. A, nobody has fully developed sexual organs as children, because that's why they're children. And secondly, pseudohermaphrodites are called that because pseudo means false, and hermaphrodite means having both male and female functional organs. Now, if you're an earthworm, you can be both male and female, get pregnant and get somebody else pregnant. But humans can't do that. There's no humans that can both produce functional sperm as well as become pregnant. So that's why they're called pseudohermaphrodites. So that question is false for two separate reasons, okay?
I'm sorry, what number was that?
That was 12.
12 in
Yeah,
27A, okay?
Yep.
All right, anybody else on chapter 27? Any of the sections, you just have to clarify what part of the notes you're in. So I think there's review questions in several different areas of 27.
For 19, what term refers to the release of an ovum? I wasn't sure if it was oogenesis or ovulation. It was ovulation.
Okay, so oogenesis is the production of an ovum, but the release is ovulation. So it is ovulation, not oogenesis.
Okay, thanks.
Uh-huh. All right, anybody else?
Can we do 35?
Read it.
What happens to sperm cells that develop in tight genes?
Okay, I didn't lecture on it, but it is in there. Remember, tight genes prevent the testicles. Well, if you read through this, it prevents the testicles from hanging away from the body. Sperm cells like to be at slightly lower than body temperature. So by tight genes pressing the testicles up against the body can cause the temperature to rise up, potentially even higher than body temperature, and you can have malformed sperm cells and potentially problems with fertility. So tight jeans or tight underwear could potentially cause fertility issues for some men who are trying to become fathers.
So would it just be A, they always die before reaching the ovum?
Oh, I
Or-
forgot you guys have choices. What's the one
Yeah.
that's closest to what I said?
They may form improper.
Yes, they may form improperly is the correct answer.
Okay, thank you.
Okay. You're welcome. Yeah, if you have choices, tell me which choice you like. I forgot that I gave you guys multiple choice options for this last chapter. Okay. Anybody else for chapter 27? All right, John, have a good one, man. Glad to have you. Anybody else? Okay. Well, I'm going to let you guys on your own then. I'm going to go ahead and ask for general questions. Anything having to do with the end of this class, the final exam, something that you think somebody else in the class might want to know based on your questions. Does anybody have any questions about what's coming up? Please remember, Thursday midnight is the very last time to take the final. Please do not wait until Thursday to take the final. Terrible idea. Take it today, tomorrow, and then take Thursday off knowing that you got the class done early. Anybody have any questions about what's coming up? Or general questions that aren't has to do with your specific individual grade? Okay, I'm gonna go ahead and stop the recording. Everybody who's watching, it's been a real pleasure, and good luck with the final.
Thank you, Professor.